Okay, we're a little late. We have one more speaker and then we will have the forum that will follow. Our next speaker, Dr. Romana-Eguia, is a scientist at the Southeast Asian Fisheries Development Center. She has over 38 years of involvement with SEAFTech and has been recently part as an adjunct professor in the biology department of De La Salle University. Her specialty is in the area of fish genetics. She obtained her bachelor's degree from the University of Philippines in Diliman, a master's degree from the Swansea University, and her PhD in the UK, and her PhD from the Tohoku University in Japan. She is currently going to be discussing the issues of genetics and immunity. Let's now welcome Dr. Rowena Romana-Eguia.
Yeah, thank you, Doc Mensch, and thank you
Yes.
for the introduction. So I hope Sir Mark can help me with the presentation.
Yes, just as a reminder, after this, we will have our panel discussion with questions. Please continue to add on your questions into the chat box.
Aquaculture. Good morning. Let me first thank the organizers for inviting me to talk about aquaculture in the new normal. I'm not a fish health expert as my field is fish genetics. Genetics is a discipline of multiple applications. Now more than ever, I'm pleased that many appreciate the science, if only because it aids us in solving current problems, COVID-19 included. Anyway, this talk has little to do with COVID-19, but it covers how the aquaculture industry is affected by the pandemic and how it needs to cope using genetic interventions in these trying times. Let me begin by saying that the current pandemic has disrupted the supply chain in the Philippine aquaculture industry. Logistics was the main problem that presented itself from when the fish spawning season started, that was in March, to date, as lockdowns continue and different means of transportation have become limited. From canceled flights to trucks that would require food passes, and even by way of your favorite Lala Move delivery, which, doesn't allow transporting live organisms. To the aquaculture sector, limited or no movement means unavailability of farm inputs such as feeds, fertilizers, fingerlings, spawners, etc., to difficulty in transporting fish farm produce to the target markets. So the pandemic has indeed adversely affected the supply, transport, and trading of fish products. But what about the effect of COVID on the fish products? BFAR has been quick to inform the public that the virus causing COVID does not itself infect aquatic food organisms and does not play a role in spreading the infection to humans. At least this is one good news. The main challenge, though, remains to be for the aquaculture industry to produce enough safe food fish for the local market. And on the whole, with or without the pandemic, the technical issues in aquaculture remain the same, and these we still continue to address. One of the main challenges in sustainable fish farming are disease outbreaks, often because of the disregard for the environment. Some fish farmers fail to follow good husbandry practices. By this we mean stocking too much fish and feeding them inappropriately or unnecessarily polluting the rearing environment with uneaten feeds. Some use stocks that are genetically poor and may even harbor pathogens. So most of the popular aquaculture commodities are known to be susceptible to parasites, bacteria, and even viruses which cause massive mortalities. Tilapias, like for instance tilapia, are known to be very hardy and resistant to diseases. And now they might be infected with the tilapia lake virus. Shrimps, on the other hand, have several known diseases caused mostly by viruses and bacteria. Two of the examples are shown here. One is the white spot syndrome virus, and the other is the early mortality syndrome, or the APEND, which is due to a toxic strain of bacteria called Vibrio parahemolyticus. Mangrove crabs are not exempt as well. Some may not manifest obvious symptoms of diseases, but these are also affected by WSSV, among others. However, of all these diseases, these are of no concern to human health, but of course, the immediate impact would be on food security and economic losses in the industry. So, how are these fish health issues addressed? For the fish farmer, the immediate remedy would be to treat the animals with some chemicals that can control the infective pathogens. And chemicals that are allowed for use in aquaculture. Another solution is basically to practice good aquaculture management in the farm itself. There are many guidelines in place, including this one drafted by the ASEAN for food fish. And farmers could follow the ASEAN GAQP to somehow avert or minimize the occurrence of diseases on farm. Another solution to the problem of disease outbreaks is by implementing strict biosecurity measures and effective disease surveillance. CFDAC AQD is promoting this to farmers, particularly for shrimp farms, given that shrimps are very susceptible to many pathogens, particularly viruses. In fact, if you may recall, for a decade from the mid-80s, the Philippines, together with China, Thailand, Indonesia, and India, produce tiger shrimps that are supplied to many export markets that include Japan, US, and Europe. With the increased incidence of shrimp diseases, which started in the mid-90s, shrimp production has not been as steady, and many countries have resorted to the production of a cheaper species, the white shrimps, which are also affected by the same diseases. Okay, so now the main focus of the paper would be the genetic interventions, and some of the genetic approaches that are used in solving fish health problems would be first, as mentioned, pathogen identification. We know that this involves genetic principles such as DNA amplification and identification of disease-causing agents based on known species-specific gene sequences. I'm talking about PCR-based diagnosis. The slide shows you the different shrimp viruses that can be identified using commercial PCR-based kits and lab-optimized protocols. The list covers all the viruses that the CFDAC AQD lab is able to identify, from shrimp disease pathogens such as the WSSV, the IHHNV or infectious hypodermal and hematopoietic necrosis virus, the Tora syndrome virus or TSV, yellowhead virus, gill-associated virus, IMNV, monodon baculovirus, whitetail disease extra small virus, the Macrobrachiomyces berginosa virus, and the enterocytozoon hepatopenae. From the shrimp viruses, we also have capacity to diagnose fish viruses from the koi herpes virus or the KHV, spring viremia of carp virus, and the VNN or the viral nervous necrosis, as well as the iridovirus. As an example of the application of a PCR-based diagnosis would be when that instance when a disease outbreak in tilapia happened in 2017 in a private farm in Bulacan. This was in fact reported in a daily, as seen in the slide. Samples were taken and analyzed by way of RT-PCR by the Fisheries Biotechnology Center, which is headed by Dr. Castroresca of BFAR, and from their analysis, the test showed positive for TILV, which was then subsequently confirmed by the National Fish Laboratory, again by BFAR, and the report of the type of the pathogen was done to the OIE. Next, another application or genetic intervention would be the use of molecular genetics in disease surveillance. So BFAR is regularly monitoring disease occurrences in aquaculture farms while agencies like CIFDEC and some academic institutions conduct studies in this regard. So one ongoing study we have at CIFDEC together with BFAR is the TILV study, or the tilapia lake virus, and we're looking at how poor water quality and culture management may lead to the occurrence of TILV in tilapia cage and pond farms. Another example of the application of molecular genetic tools and disease surveillance is in shrimps. DOST has supported a study on shrimp pathogenomics, and this involves several local agencies such as the Philippine Genome Center, Ateneo de Manila University, the Bureau of Fisheries, and CIFDEC, with funds coming from, again, DOST. So one of the outputs of this study is an online information resource on Philippine shrimp pathogens. So here they have information on the various occurrence of the viruses in the different farms. They also have a map showing where this disease outbreaks happened. Okay, so another solution or intervention of genetics in fish health management is the development of selected strains for use in aquaculture. One particular example is the development of disease-free or resistant or tolerant shrimps. So as a background, work on this started in the late 1980s in the US using the Pacific white shrimp, the Pinnaeus vanamae. And in the 1990s, it was also done in Latin America. Those countries developed the SPF, or the specific pathogen free, specific pathogen resistant, specific pathogen tolerant, and they have this term they call also unselected survivor stocks of shrimp, especially when problems that are associated with the white spot disease came about. So they developed selected strains of the Pacific white shrimp at the time that the tiger shrimp in Asia was noted to be growing poorly and have become highly susceptible to diseases. SPF refers to stocks from a population free from specific pathogens for a period of at least 2 consecutive years, and these are maintained in a highly biosecure facility. The SPF term refers to the health status of a shrimp stock, while the specific pathogen-resistant shrimp, SPR, refers to the genetic status of a shrimp stock as it relates to its resistance to a particular pathogen. Meanwhile, SPT refers to stocks that may be susceptible but do not manifest signs of the disease caused by the pathogen. All of these strains can be maintained, used, and the success in shrimp production using any of them would depend as well on the biosecurity strategy of the farm. Okay, so apart from the development of SPF, SPR, and SPT stocks using traditional selective breeding schemes, DNA markers are also used to aid selection. So of late, there are researches that are being done on the possibility of developing a TILV-resistant tilapia strain. And finally, we also see some studies on genomic selection, this time done, although these studies are quite sparse and these are done in Europe. They're developing Atlantic salmon resistant to sea lice. So it's not only resistance against viruses, but also to parasites. And they also use genomic editing as a tool for developing special strains to increase the resistance of fish to specific illnesses. But all of these are done on an experimental scale because we know that genomic editing or gene editing is a bit controversial when applied to aquatic animals. Okay. And finally, another tool that can be used in fish health management is genomics, is the use of genomics in developing functional feeds, such as immunostimulant line diets. So we have here as an example a study being done by DLSU as led by Dr. Menchi Lagman. And the intention of the project is to develop a cost-effective method in screening for potential immunostimulant sources from plants like Scotch grass, water hyacinth, red lettuce, to name a few. These plants may contain various levels of immunopolysaccharides that could help stimulate immunity in crustaceans like mud crabs when such additives are used in formulated feeds. So the general objective of the project, as you can see from the conceptual framework, is to develop two products: a set of diagnostic markers which may be used to screen candidate sources of immunostimulants, and a novel feed for the mangrove crab Scylla serrata. So I guess this is one of the last. This is the last slide, and it is hoped that there will be more undertakings in the future of this nature, so that we can help solve problems in aquaculture and improve fish production in a manner that is advanced and hopefully more cost-effective, quick, and yet precise. Thank you for listening.
Thank you, Dr. Wang, for all of that. So, wow, we had 5 very important talks, 2 from industry, and there's like information overload in my head. One quick question, Dr. Wang. Now that we all know what QRT-PCR is, and these are very sporadic events. These are unplanned for when you talk about diseases, as compared to production, for example. How do you think should investments or should a strategy be established for aquaculture, disease, and investment so that we are prepared for such events?
Yeah, so my answer to that question is that, like, we know that aquaculture in the Philippines is mostly small scale. So the first step is basically to educate the farmers on the importance of fish health management. So if they know that, and if they apply biosecurity measures, if they know where to go if their stocks get sick, BFAR offers diagnostic services from microbiological analysis to PCR analysis. Then they could bring the stocks that are infected, and they could know how to monitor and maintain, and probably cull out the sick stocks or dispose of it. The second, for large-scale producers, the major players, they can probably invest on a facility or maybe a central breeding facility that would utilize this disease-resistant stocks. It's a high investment type of thing. But if at all, maybe they engage themselves with government, private, public-private partnership. There could be a centralized breeding area for some stocks that are selected. Then it can be a big help to the industry and to somehow avert economic losses that we've seen in the past up until the present.
Having been sensitized to this issue of disease at the moment, I think much has to be done in this area.
Yeah.
So, yeah, you have a comment on that? How much is being done?
Oh, okay. So what I know is that BFAR is working, like BFAR and CFDEC are working on Bangus Fry Sufficiency Program. And CFDEC alone has invested on some biosecure facilities. That is probably just to let people know that it is important to keep your farm safe from all the pathogens that you can probably get rid of. So and also DOST is fully supportive of such endeavors. So which is why they have put in money for research. So which is why we have that mud crab thing that we're doing. Like you're leading the immunogenetics study on formulating diets. That would be the most practical approach if you ask me, because feeds can be readily be bought by farmers, and these feeds will be as effective to prevent diseases. So
it's diagnosis and prevention.